Immune system is an unique system which is efficient at recognizing the microbes, distinguishing between them, and also is able to initiate appropriate immune response in order to eliminate the pathogens. Though humoral immunity and cell-mediated immunity differ in several aspects, still they share some of the fundamental properties. Hello and welcome to SAM Biology class. Friends, today we shall discuss some of the cardinal features of immune response. These features are specificity and diversity, immunologic memory, clonal expansion, specialization, contraction and homeostasis, and the last one is non-reactivity to self. So first feature is specificity and diversity. Immune responses are specific for distinct antigen for different portions of a complex molecule like polysaccharide and protein or any other macromolecules. The part of an antigen that is specifically recognized by the lymphocyte is known as epitope. This fine specificity exists because lymphocytes which have surface receptors that are able to distinguish the subtle differences that exist in the structure of the epitopes of different antigens. Clones of lymphocytes with different specificities are present in an unimmunized or naive individual. These lymphocytes are able to recognize and respond to such antigens, and this is the basic tenet of clonal selection hypothesis. The total number of antigenic specificities of lymphocytes in an individual is known as lymphocyte repertoire, which is large and diverse. It is estimated that the immune system is able to discriminate 10 to power 7 to 10 to power 9 antigenic determinants or epitopes. The ability of the lymphocyte repertoire to recognize a very large number of antigens is due to the differences that exist in the structure of antigen binding sites of the cell surface receptors that are present in the B lymphocyte and T lymphocyte. This is known as diversity. In other words, there are clones of lymphocytes in a normal unimmunized individual where these lymphocytes have differences in the structure of the antigen binding site of their cell surface receptors. So these lymphocytes have distinct antigenic specificities. This contributes to the lymphocyte repertoire, which is extremely diverse. Second feature is immunologic memory. Exposure of the immune system to a particular antigen enhances its ability to respond again to that particular antigen. This is because of immunologic memory. Immune response to the second and subsequent exposure to the same antigen, that is known as secondary immune response, is more rapid, large and often qualitatively different from the first immune response. Immunologic memory exists because each exposure to the same antigen leads to the generation of numerous long-lived memory cells which are more efficient at recognizing, responding and eliminating different types of pathogens. These long-lived memory cells are responsible for immunologic memory. For instance, memory B lymphocytes produce antibodies that bind antigens with more affinity. Similarly, memory T lymphocytes are also more efficient at recognizing various antigen then do the naive T lymphocytes.